No, ma'am.
Yes, ma'am.
No, ma'am.
Okay, because now in real also lots of problem is there. Rumisha, how are the conditions there, Rumisha?
Ma'am, COVID is spreading a lot.
Spreading a lot, na?
Yes,
Right.
ma'am.
Okay, okay. The same thing is happening. Last time, to in real we could not observe anything like that, but this time it's becoming too much. Okay, and in Sada also the things are like that. Only Rumisha, you all are at home only all the time.
Yes, ma'am.
Uh-huh. Okay, okay. So Raj is not coming, isn't it? One is in the absent. Okay, let us begin our class. So today we will be starting with the aids. The previous all topics what we have done, they are very clear to you all. I want to ask something, any doubt in any topic, anyone? Say at least yes or no. No doubt to say no doubt.
No doubt.
Okay, the things are very clear. Did you all read all the pages of the textbook also?
Yes,
What
ma'am.
all? Okay. Devanshi, now tell me what is malt and how does it help in developing immunity? What is malt? Devanshi, can you answer? No. Rumisha, can you
Ma'am,
answer?
MALT stands
Yes.
for mucosal associated lymphoid tissue.
Yes.
The
Huh.
tissues are located in the
Huh.
tissues are
Okay.
located in the inner lining of major tracts, respiratory,
Huh.
digestive tract, and urogenital tracts, and protects the tracts from the attack of pathogens.
So any pathogens entering will be trapped in this mucosal associated lymphoid tissue and will be destroyed. Okay. So all other lymphoid organs, I hope it's very clear. What is the difference between the primary and the secondary lymphoid organ, Barbie?
Ma'am, primary lymphoids are originate and mature, and in the secondary, they migrate to the other organs and they interact with the antigens.
So what are secondary organs?
Ma'am, secondary organs are lymph nodes, tonsil,
Huh?
then appendix.
Okay, so there the remember there the lymphocytes will interact with the antigens and they will be destroying them. Okay, so that was all. Now we will start quickly with the AIDS. So AIDS you all know a lot. We have done in ninth, we have done in 10th. So whatever you know, just tell me what is AIDS. Then we will move ahead. So this year you have to study the life cycle of the AIDS children. Okay, so now
Ma'am,
tell me what is, huh?
ma'am, actually, on the WhatsApp group, I drop one message for the investigatory project topic.
Acha, acha, okay, I have not seen your in the group only. Policy
Yes,
group only.
ma'am.
Okay,
Yes,
okay,
ma'am.
I see it. Which topic is it?
Ma'am, I'm taking our fingerprints hereditary, that one for.
Okay, okay, that can be done. It is in the genetics one. Okay, then who else has selected the topic? Ritu, Barbie,
Ma'am, not yet.
not here. So when because now see only 4 days classes are remaining. You know. So then it will be difficult if you will not get the synopsis. And I'm thinking we decided that Saturday you'll show me, na? By Saturday you'll finish up Neha, isn't it? That was the time decided for the
Ma'am,
rough
for
write
yes, ma'am.
rough write up. So Umisha got the tick there. That day Neha got it, Devanshi got it. तो you have to finish it by Saturday. Okay, और Saturday को PDF भेज दीजिए रफ वाला. I will check it and I say okay. Then you can type it and keep it the soft copy, isn't it? So that way you have to do. So Shravani, then you want the synopsis just now?
Yes, ma'am. Or you can give me after class also, after completing our whole thing.
Okay, okay. So let us first complete the life cycle of the AIDS. Okay, how much you all know about the AIDS? जल्दी से बोल दो. Bittu, can you tell what is AIDS? The light is
Ma'am, it stands for
fluctuating. Say.
Ma'am, AIDS stands for acquired immunodeficiency syndrome, which is caused
Okay,
by HIV virus, which stands
okay.
for human immunovirus
Deficiency virus.
deficiency
Immunodeficiency
virus.
virus. Okay. Then what
Um,
else?
Ma'am, in this disease, small infections and minor diseases occur very frequently.
Yes,
That's all I know.
that's all. So what happens? फिर वो infection हो जाते हैं फिर क्या होता है? Neha, can you add up to what she said?
AIDS like is mostly transferred like through various. It's a
No, no.
communicable.
Transmission method हम discuss करेंगे. What is said उसमें add up करो. So what is actually happening in AIDS?
Then like what is happening after the infection?
हाँ, okay. Romisha, can you tell? Barbie?
And actually, the small disease like cough, cold, and it whenever the AIDS person suffer from it, it causes them much more effect on it, and
हाँ,
which causes them to die also.
हाँ. तो why?
And because the
Why?
immune system of there
हाँ,
is very weak
हाँ.
because of the
The
AIDS.
immune, right. The immune system gets destroyed, children. So the body will suffer from common infections. So the common infections like common cold can become pneumonia. Okay, which normally is cured in 4 to 5 days. It will last for years. Is it clear? And it may lead to pneumonia. It will become pneumonia and ultimately it may lead to death of the person also. So why does it happen? Because the immune system gets destroyed. And the immune system is of the body is not able to fight with any pathogen which is entering the body. So whatever pathogen enters the body, it will be producing the disease and it will become a serious infection or serious disease it will become. Okay, so that is the remember the immune system gets destroyed. What is the important thing which you have to remember in the AIDS? That is the immune system gets destroyed. So now I am presenting my screen, children, and mode of transmission and mode of prevention you all know very well. Okay, how it gets transmitted from one person to another, that I'll be discussing at the end. Okay, now today we will try to finish up or try to understand that how the HIV life cycle is seen in the human body, in the cell. So it is caused by HIV, which is a virus, you all know. My screen is visible to you all.
Yes, ma'am.
Okay, everyone can see clearly.
Yes, ma'am.
Okay, so now this is the life cycle of the retrovirus. Now, why we say the HIV as the retrovirus? First thing we noted down, it is known as a retrovirus because it is the virus which is having RNA as a genetic material. So those viruses which have RNA as their genetic material, they are known as the retrovirus. All have noted? Yes or no?
Yes,
Yes,
ma'am.
ma'am.
Huh. So remember, HIV is a retrovirus. Why you call it retrovirus? Because its genetic material is RNA. So the group of viruses which have RNA as a genetic material, they are all called as retrovirus. Is it clear? Now you know the genetic material actually is the DNA. What is the genetic material? DNA. And how the process or what is the role of the DNA, what is the function of the DNA, understand it expresses itself by forming a specific type of proteins. So, which proteins have to be made? That is all information, that is the information to make a type of a protein is coded in the DNA molecule. RNA will not have that information. Is it clear? The genetic material is DNA. DNA is having the information. Which information for what, Rumisha? Information for what, Rumisha? Devanshi? Neha, am I audible?
Yes, ma'am.
Okay, so what is the function of the DNA? Neha, can you tell?
No, ma'am.
Note it down then, everyone. DNA expresses itself by synthesizing proteins. Write it. Everyone write it. It's very important. The whole genetics will do all this only. ओके अभी तक आप बस इतना पढ़ते रहते थे कि डीएनए इज अ जेनेटिक मटेरियल ट्रांसफर्स इंफॉर्मेशन फ्रॉम वन जनरेशन टू अनदर ओवर यस और नो
Yes, ma'am.
हाँ सो राइट हाउ इट पासेस इंफॉर्मेशन अब ये बात को अच्छे से समझना है साल द डीएनए expresses itself by synthesizing proteins by synthesizing proteins. The information is coded in the DNA molecule. The information is coded in the DNA molecule to make different types of proteins. So different information means for specific protein there is a what they have. What the statement just now you have written?
Code.
Code, right? Okay, they have a genetic code. Okay, so specific genetic codes are there for making specific proteins. Is it very clear? Now we'll move ahead with the cycle. Once the retrovirus, that is the HIV, enters, that is human immunodeficiency virus, enters the cell. Now remember the viruses, what is the mechanism of their infection? The protein coat will not enter inside the host cell. Only it injects the genetic material. And the genetic material of the HIV is what? Rumisha, what is the genetic material of the HIV?
RNA.
RNA. ठीक है तो वो क्या करेगा पहले होस्ट सेल से बाइंड होगा वायरस बाइंड करेगा आफ्टर द बाइंडिंग it will be injecting the RNA into the host cell. So you can see here in the picture very clearly that this is the viral particle which is having the viral protein coat and inside it, it's having its genetic material, which is RNA. It will bind with the animal cell, and it will inject the RNA into that host cell. Once this RNA enters inside the host cell, remember children, it will be converted into DNA. The RNA will form the DNA. Otherwise, remember, the DNA forms the RNA. The central dogma. Now, what you do, children, quickly write down central dogma of protein synthesis. जैसे किसी भी कंपाउंड के बनाने के लिए एक फॉर्मूला होता है. The same way, for making a protein, children, there is a formula, and that formula of a cell, we call it as central dogma. We call it central dogma. Now, what is the central dogma? That is DNA. Then put an arrow, will make RNA. Okay, and this process is known as, I'll make it little bigger. So the process of formation of the RNA on the DNA is known as the transcription. But remember, inside the cell genetic material पहले डबल होता है. तो DNA से जब DNA बनता है, so you call it as, so we'll take up first the DNA. So this process, on top of the arrow, children, you write down replication. On top of the arrow, you write down replication. Then after the DNA formation, whatever information is coded there, it will be transferred to the RNA, and that process is known as transcription. So on top of this arrow, you write down transcription. Then the information which is being transferred to the RNA, it will take it to the ribosomes, and there the proteins will be formed. Ribosome is the site of protein synthesis. Everyone knows, yes or no?
Yes, ma'am.
Okay, so proteins will be formed. So this is known as central dogma of protein synthesis. What is the central dogma of protein synthesis says? That the DNA will form the DNA by the process of replication. By the process of replication. So what you have to write down on this arrow? Replication. Why you call it replication, children? Because the DNA new one which is formed on the old DNA, which is the exact copy of the DNA. It is the exactly, that is the replica of it. So you call it as replication. Then the DNA, you will find on that DNA, which process will take place? Did you all write down? Transcription. Then after the transcription, what will happen? The RNA, which is having now the information, it will be translated into protein. So you call it as translation. How many processes are involved in the protein synthesis? Or what is the formula? Protein synthesis, so we'll do it later in detail in the genetics unit, children. This whole thing we will be doing in detail in this genetics unit. Okay, but this much you remember, that how the process goes on. The DNA will form a new DNA by the process of, speak up, children.
Replication.
By replication. Then the information which is coded on the DNA will be transferred to the RNA by which process?
Transcription.
Transcription.
डीएनए के ऊपर क्या बनेगा? आरएनए बनेगा। किस प्रोसेस से बनता है आरएनए? डीएनए के ऊपर आरएनए बनेगा, तो व्हाट इज द प्रोसेस कॉल्ड? हाँ, रिपीट।
Transcription.
Transcription. Okay, now that RNA will move out of the nucleus, carrying that information, and it will go to which organelle?
Ribosome.
Ribosomes. And sorry, in the ribosomes, then the proteins will be synthesized. So, what is that process called?
Translation.
Translation. इज इट वेरी क्लियर दिस मच कि डीएनए है, डीएनए से नया डीएनए बनेगा रेप्लिकेशन प्रोसेस से। रेप्लिकेशन होने के बाद में डीएनए जो नया बन गया है वो डीएनए क्या करेगा? अपना जो उसके पास जो कोडिंग कोड्स है, जो जेनेटिक इंफॉर्मेशन है, वो ट्रांसफर करेगा आरएनए को। क्यों करेगा? बिकॉज डीएनए तो न्यूक्लियस के बाहर नहीं जा सकता। डीएनए इज प्रेजेंट इनसाइड द न्यूक्लियस, but the proteins to synthesize in the ribosome, and ribosomes are present outside the nucleus. So it needs some messenger which will carry the information coded in this DNA molecule to the ribosome. So it will make the mRNA. Which RNA will be made? Which RNA will be made?
mRNA.
mRNA. So you can write down there. Okay, so DNA will transfer the information to the messenger RNA. What is this RNA called?
Messenger
Messenger
RNA.
RNA.
I hope this all structure and that everything you have last year done properly. Are you remembering the structure of the mRNA?
Yes, ma'am.
Yes.
Yes, ma'am.
You'll have to keep those things ready because when we'll start with the genetics, so I'll say you have to revise all that and come because we'll move ahead then. What is mRNA? So mRNA will carry the information to the, say quickly,
Ribosomes.
ribosomes, and then ribosomes there इसमें सारी information coded रहेगी. And what information is coded, children? Try to understand. It is having the information that which amino acid should bind with which amino acid. Is it clear? So triplet codes are there. तीन तीन नाइट्रोजन स्पेसिस रहते हैं mRNA में. वो information उनके पास रहती है. They will identify a specific amino acid. 20 amino acids are there. This all I told you last year. The 20 amino acids are there, but the and same 20 amino acids will be forming so many different types of the proteins. That may be the enzyme, that may be any hormone. When you have done the endocrine system, you have seen so many hormones are there. And when you have done the digestive system, so you have seen the process of digestion involves so many enzymes. When you have seen the cellular respiration, there again in the Krebs cycle and all, you have seen how many different enzymes are needed for the different reaction to occur. वो सारा बनेगा कहाँ? कहाँ बनने वाला है वो सारे प्रोटीन्स, एंजाइम्स और हार्मोन्स?
Ribosomes.
हाँ, और वो इंफॉर्मेशन कौन लेके आएगा?
mRNA.
mRNA कहाँ से लेके आएगा?
DNA.
डीएनए से. ठीक है, और वो इसको आइडेंटिफाई कर पाएगा. वो इंफॉर्मेशन क्या करता है? दैट इज द इंफॉर्मेशन फॉर द आइडेंटिफिकेशन ऑफ
Amino acids.
amino acids. Is it clear? So one by one 20 में से कहाँ किसके साथ किसको अटैच होते जाना है? That way they'll keep on attaching. They'll keep on forming the peptide bond and a long peptide chain will be formed, children. And that is only a protein. So this is a central dogma of protein synthesis. What is it? What is it?
Central dogma of protein synthesis.
So in all the cells, children, this is the central dogma which is followed for the protein synthesis. But now see, we will see what happens in the retrovirus case. Is it clear? Now in the retrovirus, because retrovirus is having RNA as a genetic material, So that RNA of that virus is entering into the animal cell, that is the host cell, and now what is done from that RNA? DNA बनेगा. पहले क्या होता है, वैसे नॉर्मल सेल्स में डीएनए से आरएनए बनता है. व्हाट इज दैट प्रोसेस कॉल्ड? Say quickly, what is that process called?
Transcription.
Transcription. But here what is happening? Reverse is going on. What is happening here? Devanshi, what is happening in this HIV case?
Ma'am,
Huh?
RNA is converting into DNA.
RNA is forming the DNA. Is it clear? Otherwise, DNA forms the RNA. So, what is that process called? What is that process called? अरे वैसे ट्रांसक्रिप्शन है, ट्रांसक्रिप्शन का उल्टा क्या बोलेंगे?
Reverse transcription.
Yes. Is it clear? So in the HIV, what process takes place? Number one, reverse transcription. So keep writing step by step. The first step in the cycle is what? Infecting the host cell. Okay. Second, injecting the RNA into the host cell. Third step, reverse transcription. Are you noting the children?
Yes, ma'am.
What is the first step? Because I will give for 5 marks. This is asked. Explain the life cycle of the HIV. Very important for the board examination. Is it clear? So understand it very nicely. First, the virus particle, that is the HIV, will infect the host cell. It will bind with the host cell and will inject the RNA into it. This RNA will form the DNA. So what is that? Our first step is infection. Second step is injection of the RNA into the host cell. Third step is reverse transcription. So now the enzyme needed for that reverse transcription is the reverse transcriptase. What is it? Enzyme needed for this reverse transcription?
And reverse transcriptase.
So now from that RNA, DNA will be formed. That DNA, that is the viral DNA, that will be called as the
Viral
Viral
DNA.
DNA.
now this viral DNA will bind with the host DNA. It will bind with the
Host DNA.
DNA.
मतलब उस एनिमल सेल में तो न्यूक्लियस है, उस न्यूक्लियस के अंदर उसका डीएनए है. तो ये वायरस का डीएनए न्यूक्लियस के अंदर, एनिमल सेल के न्यूक्लियस के अंदर जो प्रेजेंट डीएनए उसके साथ वो कंबाइन हो जाएगा. तो द थर्ड स्टेप इज द वायरल डीएनए incorporates into the host DNA. Incorporates into host DNA. Genome is the total genetic material of the cell. We call it as genome. Is it clear? So it gets incorporated into the host genome. Now what will happen children? अब वो डीएनए, वायरस का डीएनए एनिमल सेल के डीएनए मतलब कि ह्यूमन सेल के डीएनए के साथ जब बाइंड हो जाएगा, तो नाउ इट विल स्टार्ट द प्रोसेस ऑफ द सेंट्रल डॉगमा व्हाट यू हैव सीन, दैट द रेप्लिकेशन विल टेक प्लेस. जैसे ही होस्ट सेल के डीएनए का कॉपी बनेंगे, साथ साथ में वायरल डीएनए कॉपी विल आल्सो बी मेड. And then what will happen? Those DNA, that is the information coded in the DNA, will be transferred to the RNA, and RNA will make the proteins which are needed for the virus particle. Which proteins it will make now? Which proteins that viral RNA will make? Speak up children, जल्दी जल्दी बोलते जाओ. Neha, did you understand?
Ma'am, I didn't get the last part.
Last part, I'm repeating. See, it got incorporated with the DNA of the animal cell. Yes or no?
Yes, ma'am.
This much is clear. Now you know animal cell में protein synthesis तो होना ही है for the different metabolic processes. Yes or no?
Yes, ma'am.
So how it is made? First the DNA will form more DNA. by which process?
replication.
रेप्लिकेशन होगा, क्योंकि वायरस का डीएनए भी इसी डीएनए से कंबाइन हो गया है, तो वायरस का डीएनए का भी कॉपी बनेगा। अंडरस्टूड एवरीवन?
Yes, ma'am.
ओके, नाउ वायरस डीएनए बन चुका है। अब वायरस डीएनए जैसे होस्ट का डीएनए ट्रांसक्रिप्शन करेगा, तो वायरस का डीएनए भी उसमें भी ट्रांसक्रिप्शन हो जाएगा, तो देन वायरस आरएनए विल बी मेड। विच आरएनए विल बी मेड?
Virus RNA.
along with the host RNA. Now, what this viral RNA will do going out of the nucleus? It will go out of the nucleus. RNA mRNA तो बाहर जाएगा न्यूक्लियस के, फिर बाहर जाके क्या करेगा, वो किसके पास लेके जाएगा उस इंफॉर्मेशन को?
Ribosomes.
हाँ, ओके, सो नाउ वो राइबोसोम किसके राइबोसोम है?
Host cell के.
Host cell के ribosome. Is it clear, everyone? So now the host cell will be guided. That is, the host cell ribosome will be guided to make the proteins which the virus wants. Is it clear? So viral RNA will guide the ribosomes to make the proteins of its interest. Understood this much?
Yes, ma'am.
Okay, now those virus proteins will be made. Those virus proteins, they will be getting assembled, and they will form the large number of the virus particles inside the host cells. और वो आरएनए फिर क्या करेगा, बहुत सारे प्रोटीन बन गया, प्रोटीन के अंदर वो आरएनए चला जाएगा और इतने सारे बहुत सारे वायरस पार्टिकल्स बन जाएंगे. So these are the new viruses. What are these? Can you see in the figure, children? Is the figure clear to you all?
Yes,
Yes,
ma'am.
ma'am.
What is happening one by one? Now we'll see it from here. Did provide my cursor is visible to everyone? Yes or no?
Yes, ma'am.
Okay. So virus going near the animal cell, binding with the animal cell membrane, injecting the RNA into the animal cell, that is the host cell. Then the reverse transcription occurs from that viral RNA. The viral DNA will be made. Viral DNA will enter inside the nucleus, will get incorporated into the host DNA, and it will start the replication. So viral DNA will be made, then the transcription will start. So viral RNA will be made. Viral RNA, by the process of transcription which are made, they will be going out of the nucleus, will come into the cell cytoplasm, and it will move to the ribosomes. Once they'll move to the ribosomes, then they will guide the ribosomes to make the proteins of their interest. Will make the proteins of, speak up,
Their
children,
interest.
their interest. Okay, and then this way new viruses will be produced inside the cell. New viruses will be produced inside the cell. Then rupturing of the host cell will take place, children. उसके बाद क्या होगा? This host cell will, what will happen? Rumisha, when will be,
Rupturing.
it will rupture. The lysis of the host cell will take place, and all these new viruses will come out of the host cell, and then they will क्या करेंगे अब वो आगे?
Acting on
Affect
the immune system.
other cells.
Right, they will infect other cells. Understood, everyone? Now I want Rumisha to explain quickly the life cycle of HIV.
Ma'am, first of all, the infected host cell will attack the human body host cell, and
Again,
then will
again, again, start again.
First of all, that um, retrovirus will attack the host cell
Huh?
and then will bind with the host cell and will
Okay.
inject RNA.
Huh?
Then RNA will be converted into DNA by the action of enzyme reverse transcriptase.
Very good.
Then the DNA will become viral DNA and will bind with the DNA of the host cell.
Okay.
Then the viral and will mm, make that host cell DNA also affected with that viral and will virus and will become new viral DNA.
No, no. Just say that once it gets incorporated, its replication takes place.
Okay,
Okay.
ma'am.
Replication and transcription will occur. What will
Okay.
happen?
Its replication and transcription will occur.
And what will be formed then?
Then viral RNA will be
Right.
formed,
Right.
and then viral RNA will undergo translation and will come to the ribosomes and will form protein of its own interest.
First, it will go out of the nucleus. You
Ha,
remember?
first
Huh.
it will go out of the nucleus and then will go to the ribosomes and will form proteins of its own interest,
Right. Huh.
and the host cell will rupture and the
Abhi kaise say? First, the new viruses will be formed.
okay, ma'am, new viruses will be formed, and when the cell will rupture, the material will come out and will also affect the other cells.
Material, what material?
Ma'am, this viral RNAs,
Viral RNAs or viruses.
viruses.
No material.
Okay,
What
ma'am.
you have to say? The
The
new viruses.
will be come out when the cell will rupture. The new viruses will uh, get out of the cell and will attack the other cells.
Will infect other cells. Okay, now try to write it everyone. That once this HIV enters into the body, which cells they will enter there. तो राइट इट ये एनिमल सेल अभी अपन ने ये जनरल लाइफ साइकिल कर लिया अब लिख लो जल्दी से दैट इट विल enter the virus H that is the HIV will enter into macrophages ये एनिमल सेल कौन सा वाला एनिमल सेल होगा बॉडी का which cell it will be what did I say just
Macrophages.
macrophages so write it okay HIV infects the macrophages then inside the macrophages then what will happen number one what will happen the first step inside the macrophages quick अरे अभी रुमीशा ने पूरी साइकिल बता तो दी सबसे पहले क्या हुआ था
Injection of the host genetic
वो
material.
एंटर हो गया उसके बाद what will happen once it enters inside
Reverse transcription.
the reverse transcription will occur and after reverse transcription
Viral DNA will be formed.
बन गया उसके बाद क्या
It will bind with the host DNA.
then
Then replication starts and more virus DNA forms, which
हाँ
then undergoes transcription to form
हाँ
virus and RNA. Then,
right
along with the host RNA, it goes out of the nucleus and goes to
हाँ
the ribosomes of the host cell,
हाँ
which and it will then guide the ribosome to form proteins of its own interest.
right
And then a lot of new viruses are formed.
that new viruses will be produced so where these new viruses will be produced where this process is going on where which cells of the body
Macrophages.
the
Macrophages.
macrophages is it clear so this way now the macrophages will be forming large number of the virus particles is it very clear now this much is very clear so macrophages are called the HIV factory what they are called
HIV factor.
HIV factor.
HIV factory. Now after this, okay, वो pass ke means the other, their new viruses will come out, they'll infect the other macrophages, then there further they'll multiply, they'll keep on multiplying. Then simultaneously some of the virus particles, these HIV viruses, will enter into the T lymphocytes. Where they'll go then?
T lymphocytes.
T lymphocytes, okay, and they inside those lymphocytes the same process will occur. Okay, the replication will occur, the transcription will occur, and they'll produce more of the progenies. They'll produce more of the viruses. Then where they will go? अब वो उधर से ब्लड सेल से अगर दे आर अटैकिंग द टी लिम्फोसाइट्स, उसमें भी अगर बहुत सारे बन गए तो वो जब लाइसिस होगा उन सेल्स का, तो वेयर दोज वायरसेस विल गो? वेयर द वायरसेस विल गो? लिम्फोसाइट्स कहाँ प्रेजेंट होते हैं?
Blood.
ब्लड में। तो जो निकलेंगे तो वो कहाँ जाएंगे?
Blood.
Blood. And then they will attack further other T lymphocytes. They will attack
Other T lymphocytes.
other helper T lymphocytes. So the process will be repeated. And you will find that this way the degeneration of the T lymphocytes will occur. Are you understanding? क्योंकि इतने सारे लाइसिस होते जाएगा, हेल्पर टी लिम्फोसाइट्स फिर लाइसिस होता जा रहा है, वो अंदर जा रहे हैं, फिर उनको डिस्ट्रॉय कर रहे हैं, आगे आगे फिर और डिस्ट्रॉय कर, तो यू विल फाइंड दैट देयर विल बी अ प्रोग्रेसिव decrease in the number of the helper T lymphocytes. Is it very clear? And that will only lead to what? That will lead to the
Deficiency of the immune system.
right, that will destroy the immune system. सारे जब lymphocyte धीरे धीरे खत्म होते चले जाएंगे, वायरस पार्टिकल सबको अटैक करता जाएगा, you will find that helper T lymphocytes will be destroyed and it will affect the immune system. The immune system will become weaker and weaker. Immune system will become
Weak.
weak. Okay, it will keep on becoming weaker and weaker. So you have done, I have discussed before, 9th 10th 11th 9th 10th में सब डिस्कस किया था कि the time taken for the manifestation of the disease is too long. Okay, the virus may have entered 15 years before in the body and the symptoms start coming or it is diagnosed after 15 years also. Is it clear? क्यों? क्योंकि ये slowly मैक्रोफेजेस में जाके फिर नंबर बढ़ाएगा, फिर ब्लड सेल्स में लिम्फोसाइट्स में जाएगा, हेल्पर टी लिम्फोसाइट्स में उनको डिस्ट्रॉय करता, उनके अंदर मल्टीप्लाई करेगा, उनको डिस्ट्रॉय करता चला जाएगा, एंड ग्रेजुअली द इम्यून सिस्टम विल बी becoming weaker and weaker. Is it clear now? It's 46. How many students have understood this whole thing? So today we have completed with the whole AIDS topic. I want everyone to draw this life cycle of the HIV in their notebook. All have understood or not?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Read the book and you will prepare it nicely. Next period, I'll be asking the questions. Okay, the life cycle of the HIV, how it enters, what is reverse transcription, how it gets incorporated, what is transcription, then what is translation. That all you have to study. And stepwise, you write it in your notebook, children. Is it clear to everyone? So now I want Rumisha Barbie to do Shanti part.